Hi students, in this video we are going to discuss the last portion of this chapter. One is a test for DNA and RNA. How can we confirm the DNA presence or the RNA presence? One of the factors that differentiate between DNA and RNA is the pentose sugar. That is, in DNA it is deoxyribose and RNA it is ribose. So, identifying these difference or a nucleic acid, if it is DNA or RNA, first of all have to identify pentose sugar component. First one is the test for DNA. Nucleic acid extract containing DNA when heated with diphenylamine in acid solution, you will get a blue color. That blue color shows the presence of these 2-deoxyribose. Because this 2-deoxyribose in the presence of an acid, it will form hydroxylevulinic acid, which forms a blue complex with diphenylamine. Hydroxylevulinic acid. This 2-deoxyribose in the presence of acid form the hydroxylevulinic acid. Then it will form a blue complexes with diphenylamine. So we can find out that this 2-deoxyribose unit is present in the nucleic acid extract, so it will be the DNA. Next is the test for RNA. RNA upon alkaline hydrolysis yield its pentose sugar component ribose. In presence of strong acid, ribose undergoes dehydration to furfural. This when heated with orsinol. Orsinol means 5-methyl-1,3-dihydroxybenzene in the presence of ferric hydrochloride gives a green color. Here you are not going to study the reaction exactly, whatever the things is going on, just to understand what we can do to identify DNA and RNA. So for first, we already see the test for DNA. For the test for RNA, what is RNA? Alkaline hydrolysis yielded its pentose sugar component ribose. In the presence of strong acid, ribose undergoes dehydration to furfural. That furfural heated with orsinol, we will get a green color. It is in the presence of ferric chloride. അപ്പോൾ ആദ്യം എന്താണ് ചെയ്യുന്നത്? ആൽക്കലൈൻ ഹൈഡ്രോളിസിസ് നടത്തും. RNAs undergo alkaline hydrolysis, we will get a pentose sugar, that is ribose. That ribose in the presence of strong acid എന്ത് കിട്ടും? ഡീഹൈഡ്രേഷൻ നടത്തിച്ച് ഫെർഫ്യൂറാൾ കിട്ടും. ആ ഫെർഫ്യൂറാളിന് എന്ത് ചെയ്യാം? ഓർസിനോളിൽ ഇൻ ദ പ്രസൻസ് ഓഫ് ഫെറിക് ക്ലോറൈഡ് ഹീറ്റ് ചെയ്യുമ്പോൾ എന്ത് കിട്ടും? എ ഗ്രീൻ കളർ. Then you can identify it is the RNA. That pentose sugar yield from this nucleic acid is ribose. So it is a RNA. Okay. Next we are going to discuss about the effect of hydrogen bonding in biological system. Molecules, that is in a biological process, the groups atoms concerned together by covalent bond. We know that what is covalent bond. On the other hand, this biomolecule assumes its characteristic conformation or interact with one another primarily through non-covalent interaction. Of these non-covalent interaction, perhaps the most important type is hydrogen bonding. That means biological molecules involved by covalent bonds. These atoms are connected together by covalent bond. These biomolecules interact with another through a non-covalent bond, that is mainly by hydrogen bonding. This hydrogen atom linked covalently to a highly electronegative atom having lone pair may get additionally bound by electrostatic force to another highly electronegative atom of the same or different molecule. This relative weak attractive force is called hydrogen bond. We already studied in the previous semester or in previous classes what is hydrogen bonding. Just that hydrogen bonding is defined here. It is the hydrogen atom linked covalent to a highly electronegative atom. Then what will happen? That electronegative atom means that will have a lone pair of electron may get additionally bonded by electrostatic force to another highly electronegative atom of the same or different molecule. Same molecule or different molecule ഇതേപോലത്തെ highly electronegative ആയിട്ടുള്ളതിനുമായിട്ട് ഉണ്ടാകുന്ന ഒരു electrostatic interaction ആണ് എന്ത് പറയുന്നത് hydrogen bonding എന്ന് പറയുന്നത്. It is weaker than a covalent bond ആണ്, but it is stronger than ordinary intermolecular forces like van der Waals forces. Okay. Hydrogen bonding can either be intramolecular or intermolecular. That already you all know about that. Intramolecular means within a molecule, same molecule. Intermolecular means different types of molecule. This electrostatic attraction is going on. Then it is called intermolecular hydrogen bonding. Both types of hydrogen bonding are commonly in the biological system. We can see that both types of hydrogen bonding. Some examples are mentioned here. Hydrogen bonding plays a crucial role in the structure of deoxyribonucleic acid. We saw the structure, double helical structure of deoxyribonucleic acid in the previous videos. There we seen clearly that the nitrogenous base are attracted, or electrostatic attraction between this nitrogenous base is by hydrogen bonding. So that double helical structure base pairing is by hydrogen bonding. That means adenine with the thymine or guanine with the cytosine are by hydrogen bonding. Adenine with the thymine, we already saw it is 2 hydrogen bonds are there, while in the case of guanine and cytosine there are 3 hydrogen bonds present. That these type of hydrogen bonds hold them together to a helical nucleic acid chain. That helical structure is the helical structure of this nucleic acid is mainly by this hydrogen bond. Secondary helical structure of proteins. Here we first we discuss about nucleic acid structure. Then second one is the protein alpha helix structure that we will see in the next chapter. How this secondary helical structure of protein. That in that protein secondary structure also that is derived. That secondary structure is derived from the folding and coiling of polypeptide chain. This fold or coiled structure are held together by intramolecular hydrogen bonding. You know that in a DNA that is an intermolecular hydrogen bonding, but the secondary helical structure of this protein is by the coiling or folding of this polypeptide chain. Due to that coiling and folding, there will be an intramolecular hydrogen bonding between CO and NH of the peptide linkage. Secondary beta structure of a protein is also derived from the hydrogen bonding between long polypeptide chain that lie side by side in a zigzag manner. Beta structure in different polypeptide is attracted through this hydrogen bonding a zigzag manner that you can see the next chapter. You can see in the next chapter. Hydrogen bonding plays an important role in deciding the tertiary structure of protein also. The tertiary structure of this protein also is deciding by this hydrogen bonding. So first in the nucleic acid, second in the protein, we saw that hydrogen bonding. Next one is the cotton is a pure cellulose. You know that cotton is a pure cellulose, which is a linear polymer of beta glucose. Cellulose means it is a linear polymer of beta glucose. These linear polymerized chain of cellulose are held together by hydrogen bonds between each group of glucose units of adjacent strands. In this cellulose, these beta glucose units are held together by hydrogen bonds between each group of the glucose unit to the adjacent strands. Okay. So, in these three, we can see the hydrogen bonding presence in biomolecules. Nucleic acid, protein and cotton. Okay, thank you. This chapter is finished. So, if you have any doubts, you can ask, and I think there is no need for a Google Meet because it is a biological chemistry, biochemistry. So this all you maybe studied in your zoology class or in your sub microbiology class or anywhere. So thank you very much.